Mockingbird is a closed-loop neurofeedback system that's being developed at UCLA with funding from the National Science Foundation. This system can process neurophysiological recording data in real time to detect events in the brain and instantaneously trigger neural or sensory feedback. Here are EEG signals being recorded from the brain of a freely behaving rat. You can see several kinds of events in this data, including theta rhythms, sharp waves, and ripple events. Offline methods for detecting these events rely upon acausal filters that are not appropriate for triggering instantaneous feedback. Mockingbird solves this problem using machine learning technology. Users can train neural networks to function as causal online filters that closely mimic the performance of offline filters, but without incurring any filter delay. Feedback stimulation can be triggered by detection of transient neural events such as sharp waves, or synchronized to a specific phase of ongoing neural oscillations. Mockingbird can process hundreds of recording channels simultaneously. The system is available as an application for the Neuralinks hardware processing platform. It will become available for other data acquisition systems as well.
I'm Jacob Robinson. I'm an assistant professor of electrical engineering at Rice University. My lab is actually working at this interface between electrical engineering and neuroscience. And what we like to imagine is creating technologies that can be implanted inside the body, made so small that they barely interact with the biological tissue. And they can be distributed in the brain and throughout the rest of the body. And what they'll do is they'll sit there and they'll monitor activity to try to understand what's happening normally in healthy individuals. And then to give stimulation capabilities to these same kinds of devices that start to restore function both to the brain and to the body in a natural way that's smart, that feeds back on the signals that it's recording. And the hope is that with these kind of tiny stimulators that can be implanted in the body, we can start to treat disorders that might be related to pain or depression that are resistant to traditional pharmaceutical treatments. One of the things that I'm particularly excited about is the idea of using magnetic fields as a way to interact with those cells. The reason why I think magnetic fields are really important for these kinds of applications is that magnetic fields penetrate our body very easily. Now the challenge is, can we use these magnetic fields to deliver power to microscale devices that can stimulate and record brain activity? One of the wonderful things about the Brain Initiative is that they've been able to fund engineers that are working at this interface with neuroscience and neurotechnology who might not normally be pushing their research in that direction. And then to connect these types of engineers with the scientists who are asking the questions. Thanks to the funding and support that we've had from projects in the Brain Initiative, we've now been able to take some ideas and translate them into devices and technologies that are beginning to make their way into the hands of our collaborators in neuroscience and our clinical collaborators at the medical center. The Brain Initiative is really open-minded about supporting ideas that might be outside your comfort zone and try to find support from collaborators who will help fill those gaps. For people who are considering the Brain Initiative as a way to support their work, I would probably give them 3 pieces of advice. Be bold, reach outside your comfort zone, and talk to people. Talk to lots of people who are outside your discipline. So go ahead and put your wildest and craziest ideas out there.
An important application of virtual reality is that we could potentially use it to enhance your knowledge of reality. My name's Dr. Arne Ekstrom. I'm an associate professor of psychology at the University of Arizona.
My name is Dr. Derek Huffman. I'm a postdoctoral fellow working in the Human Spatial Cognition Lab.
We try to understand how people navigate space and how people remember where they are.
In our experiment, we wanted to test between two competing hypotheses. The first idea is that body-based cues are fundamental to how we represent space. The second hypothesis would suggest that body-based cues don't matter as much once the environments are well learned.
Our study involved a large-scale environment that required exploration and looking around at many different angles. We had 3 different conditions.
In an enriched condition, we had participants freely navigate on the omnidirectional treadmill that will actually allow people to turn and walk in any direction. In a limited condition, participants were able to freely rotate their bodies and heads around, but then they move forward using the joystick. In a third condition, which we call the impoverished condition, participants use a joystick for all movements throughout the environment and were instructed to keep their heads and bodies facing forward. After participants learned each of 3 cities, we then imaged their brains using a technique called functional magnetic resonance imaging.
We had them retrieve information about the relative directions of landmarks that they had experienced when they were navigating. What the study suggests is that body-based cues with sufficient exposure don't make any difference for how we remember and how our brain codes spatial locations. Perhaps just giving them the visual information via head-mounted display could be sufficient.
Hello, I am Pepe Contreras Vidal, Director of the Brain Center at the University of Houston. This is a collaboration with Arizona State University and industry to develop, design, and validate new technologies to help people with disabilities. The long-term vision of the center is to become an international hub for research and education at the intersection for art, science, engineering, and society. Some of the projects that are currently funded by the Brain Center relate to concussion, invasive and non-invasive technologies to listen and write to the brain, and also educating general public. The center is also involved in outreach activities. We work with children at museums collecting data in action and in context, and we collaborate also with the arts and the humanities to understand creativity and neuroaesthetics.
And we use laser-based diagnostics to non-intrusively make quantitative measurements of what's going on in the flow. Really are able to do types of measurements that aren't done in too many places in the world, so this really is a state-of-the-art facility. We do a tremendous amount of collaboration with universities across the country. Right now, I'm the lead PI and CU is the lead institution on a very large grant from NSF, which is trying to understand how animals navigate through odor plumes. And so what we're doing in our lab here is we're creating digital landscapes of what odor plumes look like, and then that data is being used by other groups, which are mostly med schools, neuroscientists, computational
This team
neuroscientists.
measures these odors underwater. They start by pumping 5,000 gallons of water through a 50-foot-long tank. Next, they spread laser light into a sheet that slices through the water, and then finally comes the dye.
And so what we're doing is we're using a special kind of dye as a surrogate for the odor. And this dye has exactly the same types of properties that an odor would have if it were in this flow. But it has a special property that if we shine a laser light on it, then it fluoresces.
Vision is a coordinated effort between the eye and brain. When our eyes see an object, The light from its surface travels to the retina, where it is transformed into neural signals. These signals travel through the parts of the brain dedicated to vision. In the primary visual cortex, the brain represents the image in a map that matches the visual world. Our project proposes one potential idea to take advantage of this mapping so as to create a new prosthetic that will allow blind people to see. But the visual pathway will be different for a person who is blind. Here, the patient will wear a pair of eye-tracking glasses that monitors what is happening in the visual scene. These glasses will also track exactly where the eyes are pointed so that we know which information to transmit into the central retinal part of the brain. To get the information from the camera into the brain, we will first use gene therapy to turn specific neurons in the visual pathway into photoreceptors that take the place of the damaged retinas in the eyes of a blind person. Now we can stimulate those neurons with light. And our preliminary data shows that stimulation of these newly created photoreceptors drives large neural responses in the visual system. Here is an overview of how our optogenetic brain system, or OBSERVE for short, will work in patients. Visual information that is centrally fixated by the eyes in the visual scene will be transmitted from the camera through ultra-wideband encrypted wireless telemetry to the OBSERVE implant. This suggests that if we beam in light in the correct prosthetic pattern, these neurons in green from the brain's visual cortex will respond like they do to real visual inputs from the eye. But we can't just stimulate the brain without continually measuring its response. Why? The signals that the neurons send onward to the visual cortex, flashing in green, can vary in intensity in our system with each firing. We need to monitor in real time for these variations and calibrate them to make sure the signals sent onward to the visual cortex look like real vision. Here's how we will calibrate our system, and it takes some scientific innovation. First, remember my earlier mention of the gene therapy that turned neurons in the brain into photoreceptors? By also using gene therapy to fill neurons in the visual cortex with bioluminescent proteins of multiple colors, we can make these neurons blink like a rainbow starburst of fireflies whenever they are active. Here, you can see over 61,000 of these labeled neurons in the largest microscopic image ever made inside the brain. Therefore, we will read out the brain's response in our patients by transfecting these bioluminescent calcium indicators into the visual cortex. Second, we will need to create a brain implant that contains both a video projector and a color camera. It will beam in the eye-tracked video information from the scene cameras on the patient's glasses and also read out the bioluminescent brain activity to make sure the signaling remains calibrated at all times. OBSERVE will be surgically implanted over the visual cortex so as to beam the scene camera video feed to the genetically transformed photoreceptors in the brain, which will stimulate visual perception with naturalistic synaptic transmission.